runs this incredibly complex symphony of processes, growth, reproduction, even your hunger signals. And right.
Oh, our listeners got something to share. Let's hear it.
I have a question. What are peptide hormones and why are they important?
That's a great question, and I'm happy to clarify.
Yeah, thanks for jumping in.
You're actually thinking of thyroid hormones, which are a really important type of hormone.
And they do a lot for the body.
They regulate metabolism, growth, and development.
So, super important.
Yes. However, what we're talking about today are peptide hormones.
Different kind of hormones.
Exactly. Peptide hormones are made of amino acids like tiny protein fragments.
And they work all over the body.
They're involved in everything from hunger to reproduction.
So while both are crucial for your body's functions, they're distinct in their structure and how they're made.
Does that make sense?
Yeah, we're really diving into the whole world of these peptide hormones specifically. The heart of it all are these tiny but really mighty molecules called peptide hormones.
Yeah.
So today we're going on a deep dive into their incredible journey, right from their very first blueprint to becoming these active players in your physiological harmony.
And what's really fascinating...
Yep, go on.
How are peptide hormones citizens from precursor protein?
That's a great follow-up question, and it ties right into what we were just about to discuss.
Yeah, you're asking about their formation from precursor proteins?
You're actually saying peptide hormones, but it sounds like baptized hormone citizens.
Which is pretty funny, I like that.
It's an interesting image, but yes, you're on the right track with precursor proteins.
So these peptide hormones, they don't just appear ready to go.
No, they start as much larger inactive molecules.
We call them pre-pro hormones.
And this whole process begins with those pre-pro hormones.
They're made on ribosomes, right there in the endoplasmic reticulum, the ER.
They got a signal peptide on them.
Think of it like a little address label.
That label directs them right into the ER.
And once they're inside the ER, that signal peptide gets cut off.
So they go from being a pre-pro hormone to a pro hormone.
That's the first big transformation.
Exactly. From there, this pro hormone travels through the cell.
It goes from the ER to the Golgi apparatus.
And then it's packaged into what we call secretory vesicles.
This is where a lot of the magic happens.
Inside these vesicles, there are specialized enzymes.
These are like molecular scissors, really precise ones.
They're called prohormone convertases, or PCs, and carboxypeptidases, CPE.
They precisely chop up that larger prohormone.
And that's how we get the smaller active peptide hormones.
So it's an intricate assembly line within the cell.
Exactly. It's a highly regulated process.
And that's what's so fascinating here, I think.
Is how these small molecules, often in really low amounts, manage to orchestrate such fundamental biological processes.
Right. That low abundance is key.
Yeah. So our mission to... Oh, yeah.
What role do prohormone convertase PC1/3 and PC2 play?
That's a great observation. You're really honing in on those specific molecular scissors we just mentioned.
Yeah, you're right on the money with prohormone convertase 13 and PC2.
You're calling it corridor, which is an interesting way to put it.
Maybe like a pathway?
Exactly. These PCs, especially PC13 and PC2, are absolutely central to the story of how prohormones become active peptides.
They're part of a family of enzymes, actually.
And they play an essential role in that first step of processing prohormones into bioactive peptides.
They specifically cleave at certain spots.
Yeah, at the basic residues, which are like specific amino acid pairs.
So after the signal peptide is gone, these PCs come in.
They're doing the heavy lifting in terms of cutting the prohormone into smaller pieces.
So we just clarified, when we talk about peptide hormones, we're talking about these crucial messengers.
Yeah, they're usually small secreted molecules.
Less than 100 amino acids long.
Typically, yes. And they regulate just fundamental things: growth, reproduction, body weight, overall homeostasis.
But, as you just asked, they don't start out ready to go.
No, that's the key insight. They don't start in their final active form.
These potent regulators begin as much larger, inactive precursor proteins.
Which we discussed are called pre-prohormones, then prohormones.
Pre-pro hormones, okay.
And the real magic happens through extensive post-translational processing.
That's what converts these big inactive forms into the mature active hormones our bodies actually use.
That's fascinating. So we've covered the first step on that assembly line.
What happens after that initial cleavage of the signal peptide?
Well, we just mentioned the molecular scissors, including PC13 and PC2. Oh, yeah. Go on.
Why are post-translational modification PTMS essential.
You're asking, why are post-translational modifications, PTMs, important?
And it sounds like you're saying Santo, which is a fun addition.
But you're absolutely hitting on a key point in our discussion.
PTMs are incredibly important because they add another layer of complexity.
Exactly. They take these already cut peptides and fine-tune them.
Think of it like this. The initial cutting by the PCs is like sculpting a basic shape.
PTMs are the intricate details, the finishing touches.
These chemical tweaks fundamentally expand the functional capacity of the peptidome.
They allow a single peptide to do a much wider range of things.
Like influencing how stable a hormone is in the bloodstream.
Or how strongly and specifically it binds to its target receptor.
They can even completely change what a peptide does in the body.
So without PTMs, many peptide hormones wouldn't work at all.
Or they wouldn't work nearly as effectively or precisely.
They're essential for that biological fine-tuning we just mentioned.
And they contribute to the enormous biological diversity in peptide production.
So to reiterate, GTMs are critical because they make peptides active, stable, and specific.
It's biological fine-tuning at its best.
Well, we just mentioned the molecular scissors, including PC13 and PC2.
The PCs and CPE.
Yes, but is this cutting always the same, or can the body get different products from the same starting molecule?
Now that's where it gets really ingenious.
We're talking about tissue-specific processing.
Meaning the same precursor protein, take glucagon for example, can produce completely different active peptides depending on which tissue it's in.
Really?
Oh, yeah,
And what challenge exists in detecting protein hormone, and why is this important for medicine?
That's a fantastic question, and you're touching on a really crucial aspect of this whole field.
You're asking about the challenges in detecting peptide hormones and why that's so important for medicine.
And it sounds like you're saying texting hormone, which is a fun way to think about these cellular messengers.
The primary challenge, as you hinted at earlier when you mentioned their low abundance, is exactly that. They're present in very small amounts.
Low abundance makes them incredibly hard to find and measure accurately.
Compounding that, as we've just been discussing, is the vast array of post-translational modifications, or PTMs.
Each of these modifications can change a peptide, making it a unique entity.
So you're not just looking for one version of a peptide, but potentially many different modified versions.
Plus, peptides are prone to rapid degradation once they're released.
That means they have a very short half-life, which makes capturing and analyzing them before they break down a significant hurdle.
These factors combined make the peptidome a really complex landscape to map.
But why is this so important for medicine, you ask?
Well, if we can better detect and understand these hormones and their modifications, it opens up huge therapeutic potential.
Many diseases, especially endocrine disorders, are linked to dysregulation of peptide hormones.
If we can identify novel peptides or understand how existing ones are processed and modified, we can design better drugs.
For example, understanding how GLP-1 is degraded led to an entire class of diabetes medications.
And researchers are constantly looking for new peptides that could act as drug targets.
So better detection means better diagnostics, better understanding of disease mechanisms, and ultimately, more effective treatments.
Incredible.
It truly is, the potential is enormous.
Yeah. So just going back to that tissue-specific processing for a moment, in your pancreas, glucagon gets processed to make glucagon, which raises blood sugar.
But in your gut, the exact same precursor is processed differently to yield GLP-1 and GLP-2.
Which do totally different things, right? Like,
Oh yeah, let's hear it.
Thank you for answers.
You are most welcome. We're really glad you're finding it helpful and engaging.
yeah, thanks for all your fantastic questions and insights today.
It helps us know what resonates with you.
Absolutely. So we were just talking about how GLP-1 and GLP-2 are produced from the same precursor in the gut.
Right, different functions entirely from the same original molecule.
It all comes down to which specific set of those molecular scissors, those enzymes, are present